Good morning, happy Tuesday, and welcome to Chapter 11. Unfortunately, we're not going to be able to watch some great movies together, and sadly, I think this is also the only time we're going to actually see pandas. Sorry about that. Today we're starting reproduction, which means we are going to see the basics of reproduction in the cell cycle, find out what chromosomes and genes are, and talk about simple cell division. I'll give you a sec to check out this classic Far Side cartoon at the bottom here, too. By now, we hopefully all know that DNA is the instructions for life. I think this has been the first slide in pretty much the last week and a half for the stuff. It's also what we call the hereditary information, and it stays in the nucleus. We also know it's a huge molecule with a twisted double helix shape, but it's not actually just one long chain of nucleotides. It's not just this enormous, single double helix molecule. The DNA in your cells actually exists in 46 different smaller pieces called chromosomes. So instead of being one gigantic molecule, DNA is just divided up into some smaller pieces, when it makes it a little bit more manageable. So this way it's easier to read the instructions and do cell jobs in the same way that an encyclopedia might be divided up into one book for each letter of the alphabet, rather than putting all information into one enormous volume. Hopefully you actually understand what I mean by that, because I know encyclopedias are probably not the most common thing anymore. It's just we have something called the internet. But just go to a library, and I really hope there are still encyclopedias out there, because otherwise, what are we even doing? So we've also heard this word genes, and these are the individual recipes for proteins we've seen a little bit about recently. And these are found in specific spots on each chromosome. So chromosome is really like just the physical twisted form of the DNA when it gets really kind of tightly packed and condensed, and the genes are specific spots on those. So this is roughly what a chromosome looks like here. This would be a chromosome right here, and this would be another chromosome right next to it. And you can see they have sort of these two sections. It sort of pinches in the middle right here. Usually you see that. The chromosomes will typically be, sort of stick or rod shaped like that, and then usually have some kind of area, maybe a little bit close to the top, bottom, maybe it's a little bit more towards the middle, but usually it pinches in a little bit at some point to give it that sort of two-sectioned look that you see here. Now, sometimes two chromosomes will actually sort of hang out together and almost lay across each other in an X shape, which is actually what we see back here. You see our double helix right here, and it just kind of twists up. If you know the model I have in the classroom, it's not always out like that with a double helix. You can see the inside bases. Sometimes you can sort of press it down and it gets sort of compressed, and it gets more of like a really sort of tight ribbon or string looking structure, and that sort of twists around here. As you can see, it wraps around these little circular looking proteins, and that just sort of gets tightly packed into this really finely wound ribbon or finely wound rope. And see, it keeps folding up into even more of this ropish structure. And it keeps folding and folding and folding, and that just then gets packed tighter and tighter and tighter into this thing finally over here that we call the chromosome. So this will really be like one chromosome right here laying across a second chromosome right there. So they take on sort of an X shape. And the important thing here is that genes are not in random places on chromosomes. So for these two examples of these two chromosomes, we say maybe these two genes right at the bottom are the eye color. So the eye color gene is on this person's chromosome, the exact same same places on this person's chromosome. Maybe hair color is this gene right here. The chromosomes are always in specific locations. So if we want to know what someone's gene is for a specific trait, we now at this point know where to go and look for that thing. As the old saying goes, just don't trust potatoes. That's an expression, right? I think. So as I was saying a second ago, a lot of times the DNA is just sort of unfolded in this traditional double helical shape that we know, because that makes it really, really easy for those little enzymes we've seen in the last chapter to go in and read what these letters up here actually are to make the copy, make the mRNA, make a copy of the DNA if you need to, or send a transcript out to the ribosome. But other times you really don't want the DNA all kind of unwound. You want to keep it nice and safe. And at that point, all these bases are just kind of move in and slide together. We kind of fold up into this more tightly wound shape. And as you can see, DNA just gets really insanely, extremely tightly packed and wound. So if this now is this really kind of condensed DNA string, it eventually just gets kind of wrapped around like a little spool of thread on these little round protein pieces. And then this now is that same thing we just saw up top. And these little strings that look like almost pearls on a thread, they now get sort of wrapped around each other, and that now would form this structure, and then this whole twisted shape of those will get wrapped around again, and it folds up in other directions. And again, it's just sort of this twisted roping wiring shape, and that just now becomes this huge jumble that we call the chromosomes. A chromosome is really just this insanely tightly packed, powerfully condensed form of the DNA that, if at times you do need to kind of have the DNA in one place where you know where it is and not just this crazy unwound spaghetti looking structure, it's really easy to do this, and the chromosome makes it very, very manageable to do stuff with. You may notice a YouTube link on a previous slide at the bottom. I fully encourage you to go check that out if you're having any troubles just kind of visualizing or picturing how this chromosome folds up, how the DNA kind of condenses to itself. I'm going to try to put the video in here, at least provide a link, maybe even in OneNote. If it won't properly save the PowerPoint, it might be a little bit long to do that. But in any case, that's a really helpful, it's only a 1.5-minute video. I think it's very good to just be able to visualize everything. It's nice computer animation that will probably help you. So at this point, I'm going to throw it back to something we talked about when we looked at DNA fingerprints last week, which is that everyone's DNA has to come from one of two sources, and that's their parents. So you either get your genes from mom or your genes from dad. I told you in the previous part of this thing today that every human cell has 46 chromosomes. Half from each parent means that we get 23 of those 46 from mom and the other 23 of those 46 chromosomes from dad. We can see that in this picture here. So this is a computer colored picture that we can, just to kind of help us see things better. But notice that you have 2 copies of each chromosome type. So they're numbered up top there, or right below the chromosomes. So here we have 2 copies of chromosome number 1, 2 copies of chromosome number 2. And you can see they kind of have similar shapes. They're roughly the same size. They're not all identical, so not every chromosome looks alike. So you can see down here, chromosome 21 is pretty small, where chromosomes 1 and 2 are pretty big. But notice we have 2 copies of each thing. And we now call these homologous chromosomes, or homologous pairs. And this term just means that both mom and dad each give us a copy of every chromosome. So we now have 2 copies again of chromosome 1, one from mom, one from dad, 2 copies of chromosome 2, one from mom, one from dad, 2 copies of chromosome 15, one from mom, one from dad, and so on, up to all the way here, chromosome 22. Now, if you're doing the math for this, that only gives us 44 total. 22 chromosomes times 2 is only 44, not 46. So the last 2 chromosomes anyone has are the sex chromosomes. So that means you have 2 copies of an X chromosome if you're a female, or an X and a Y chromosome if you're a male. But those will give you the 46. 22 times 2 is 44, plus either XX or XY gives you 46. And again, just notice that with these homologous chromosomes, you have the 23 different types, and half from mom, half from dad. Each parent will give you a copy of one. And where I said before how the genes are always in specific places, that is definitely true right here. If we look at mom and dad's chromosomes to compare, you'll see that they both give you a copy of gene A right there. Maybe again that's the eye color gene, and you would have had a little a here, so you're getting the same type of gene from mom and dad. Maybe they both have blue eyes. The next one down here, we see gene D, and it's in the exact same place. There's a big letter D over here, a small letter D over there, so it's a slightly different form. Maybe mom has one color hair, dad has a different color hair, but it's the same type of gene they're giving you, and that type of gene is in the exact same place on each chromosome. That's the important thing here, that the genes are always very well localized and not just randomly placed on any given chromosome. If you remember ever learning about butterflies, you know exactly what a life cycle is. A caterpillar eats and grows, eventually it forms a cocoon, and emerges as a butterfly. This is a step-by-step regular process that's well regulated. It's not just, again, a random set of events. And cells do the exact same thing, so that one cell can split and become 2 different cells. Most of the time a cell is alive, it's really just going about its business, doing its cell things, and we call that interphase. So really nothing special is happening. It's growing, it's doing whatever jobs it needs to do, depending what kind of cell it is. And at this point, the DNA is going to be unwound in that kind of looser double helix shape. So again, you can get to the genes, you can make copies of them, you can send them out as mRNA to the ribosome, and so on. Interphase then is divided up into 2 growth phases, these G phases, G1 and G2, where again, the cell is just actually growing. So it starts as a smaller cell, just as a person starts out smaller, it grows, it kind of gets into its role and its different jobs it has to do, and does whatever it has to actually carry out as a living thing. In between those 2 times is what we call S phase. The S stands for synthesis, or making something. And this is when a cell actually makes a full copy of its entire DNA, which we saw last chapter. That's where the ladder kind of unwinds, opens up, and the new bases come in and just sort of add pieces to it to make 2 identical copies to each other and to the original. Now, once this is done, once the DNA is fully copied, the cell goes back again to do another growth phase in G2. Again, just sort of goes through the processes, kind of gets ready, replicates some of the organelles, gets ready to divide. until it gets to the point where it is fully set and ready to divide and make a new copy of itself. And that's this process now that we call mitosis. You may have heard that word before. I know I mentioned this to some of you today in Extra Help, and it seemed like you've heard this word at least at some point. The copies of DNA at this point will be split up between the two new cells that are being created so that everyone gets the right amount. It's not that one cell is going to get a little bit and the other one gets all of it. Everybody's going to get the exact correct amount of DNA because you made a copy already. You have to always copy your DNA first, or you can't ensure that the cells are going to get the right amount. Once that happens and you get one cell perfectly dividing into two new cells, this whole thing is just going to start over. So those new cells will start to grow, and eventually at some point in their life cycles, they'll have to make new copies themselves, and they'll divide their DNA and then go back to work and grow a little bit more, and then divide and keep going, everything like that. We will get into mitosis in a few days, but the general idea of it is as follows. Asexual reproduction is one cell reproducing by splitting in half. This isn't like when animals have two parents to make offspring. Asexual reproduction has what's known as a parent cell that divides perfectly in half to make two identical daughter cells. Just like when the DNA copies itself and the daughter strands are made, same thing here. The daughter cells are identical to each other and to the parent cell. So basically the parent is becoming two children. It's not like what we would think of as animals. Again, this is not like sexual reproduction when they're two distinct parents and children. In this type of reproduction, the parent essentially does become two different children by splitting into two identical parts from what it was originally. Just to be clear, human beings reproduce sexually by having children who get some DNA from each parent and are a totally individual and separate combination of genes. But we also use asexual reproduction at every moment of our lives. We use asexual reproduction to make new blood cells when we are growing from individual sperms and eggs first fertilized into a human being, to repair damages or injuries in our bodies, and to make more cells that will increase our size and mass, letting us actually grow. What do you think some pros and cons are, advantages and disadvantages of this type of reproduction? If we're thinking about pros, this is going to be really fast, right? You don't have to wait for two parents. It's not a elaborate process where you have to find any kind of mate. If a cell suddenly needs to reproduce and divide, it can go to it really pretty quickly. It just has to get everything in order, and then all it has to do is actually split. This can be a very, very fast and efficient process if it needs to. So you can very quickly increase your population size. If you were, say, a bacterial cell, sometimes that's not really good. We don't typically want bacteria to create more and more of themselves that quickly. It can be really bad for us. But if you are a living thing that reproduces this way as a single-celled organism, that can be pretty good. And for us, again, it also helps us grow and heal in really quick ways where we don't have to wait a long time to suddenly heal up a cut or something like that. How about a con? What would be some cons? For this, I would look at something we'll see a little bit more going forward, but you're going to have the same DNA. So you're not really getting a lot of variety. You're not really getting any advantages. You're not changing. You're not advancing at all. It's just the same old thing. That can be kind of boring. That could actually be very limiting. And it can also really be damaging when we start talking about antibiotic resistance and things like that. In terms of living things, not really just our cells in our own bodies, this can be bad if you're trying to kind of increase variability and try to stay alive in a changing world around you. When simple asexual reproduction takes place in bacteria, so when it's just asexual reproduction of an entire organism that happens to be single-celled, it's called binary fission. Binary meaning two, fission meaning to break into pieces. So check out this bottom picture. The DNA is copied first before anything happens to the whole cell. Right here we see one chromosome DNA. It's not a lot of DNA because they have a very small, simple cell. And it will now, oh, my ink got messed up. It'll now be made a copy of. Why won't that let me erase it? Who cares? It'll make a copy of the DNA right there. So now you have two little rings of DNA before anything actually happens. This has to happen all the time. All these processes we're going to see in this chapter, any kind of reproduction, you always have to copy your DNA first, because if you simply try to just make a new cell, rip your cells in half, divide, before copying your DNA, something's going to be lost. Somebody's going to get too much DNA, somebody's going to get not enough DNA. You need to make a copy of the DNA first so that you definitely have enough to go around. One way that living things use asexual reproduction on a smaller scale is to replace cells and heal injuries. If we get a cut, we use asexual reproduction to replace our cells and make copies of damaged or lost cells or lost tissues to make a scab and then heal us up or again to grow. But other organisms can use a process called regeneration. This is when not just individual cells, but entire body parts can be replaced or regrown inside of a body. So we see this in this picture, how a worm called a planarian can be cut in half and the lost pieces replaced. Actually, planarians are similar to earthworms and starfish, and that cutting off a piece lets you make multiple copies. A severed starfish arm or planarian tail regrows in different directions to make an entirely new organism. Don't go and cut earthworms in half, but if an earthworm is damaged and everything is kind of maintained, it's not just totally crushed or anything, the earthworm halves can then regrow into 2 new earthworms. You see the exact same thing here with this planarian. We think of more of the head area and the tail area here, but if we cut it right down the middle, this now head part grows in a new planarian, and the tail will now actually sprout a new little head there to become a second totally new planarian. Same way that we see this starfish over here. The severed arm of the starfish will now grow into an entirely new starfish. All the information is there in its cells. It just needs to actually put itself together again. That's a pretty cool trick, right? We can see how this little newt lizard here is able to regenerate when injured. So that's lost limb regrows in not too much time. You see in the span of, what, about 9 weeks or so, if the newt undergoes some sort of injury here and just has this kind of little stub where its arm used to be, say it was damaged in some sort of struggle, or maybe a predator hurt it some way, within a couple of weeks it now goes to more of this little extended area looking a lot more like an arm, and these little stubby fingers will grow, and the cells in here between the fingers actually start to die out to make a little hand or paw looking shape to it, and eventually gets back normal. Unfortunately, we as humans are not able to regenerate. Some parts of our bodies can regrow, and there are some exceptions in certain situations where damaged pieces of certain organs can be replaced over longer periods of time. But for the most part, this really just doesn't work for us, which is kind of a shame because this would be a really great thing for people to have in a repertoire, I think. That would kind of be neat, I guess. But sadly, again, human trials just never really seem to quite work out too well. Last thing for today. Normally, asexual cell division works like this. One cell splits into 2 at precise times and only then. Signals are sent to make the cell divide, and that stops dividing until it's told to do so again. So if you get a cut, your cells know to keep making more and more cells just long enough to patch that hole up, get you back to normal. Maybe you replace any blood or skin that was lost, and then everything shuts down and goes back to whatever it was doing until maybe something happens in the future and needs to do that same kind of process again. If something does eventually go wrong, like we see happening right here, where we see this where it says cell damage and nothing is repaired, the cell will try to fix the problem, and if it can't be corrected, if it's just sort of beyond that point where anything useful can be done, the cell will actually kill itself by a mechanism we call apoptosis. This word right here, apoptosis, that's called programmed cell death, or the cell suicide term. And that's where the cell basically just hits the panic button and says, nope, we're going to lock this down from the inside, and it'll actually empty all these little digestive enzymes it uses to break down other molecules into the interior of the cell and kill itself to stop any potential problems from spreading anywhere else. A really, really helpful thing that happens a lot in our bodies to stop bad things from going around. That's how it should work normally. Sometimes things can go really badly though, and the cell doesn't recognize a problem until it's too late. So maybe there's an issue where the cell doesn't see something as a threat, or it could be that some signals are incorrectly sent, where a cell begins to divide and never actually stops. It's told to divide at the wrong time, the wrong place. It just keeps going on and on unchecked, and these are all elements of cancer, which is an uncontrolled growth caused by continuous cell division that just never gets told to shut off. So it should have stopped here. Usually some kind of mutations you see there behind it, and it just keeps dividing and dividing and dividing, and on and on and on, and nothing tells these cells to stop. And because they're your cells, technically, your body doesn't know anything's wrong and just figures, oh, we're going through a growth spurt, and never shuts this process down. So we can see this up here, normal cells here, and then we get some kind of abnormal growth. It could be some little mutation. Sometimes mutations don't really lead to anything. Abnormal cells, they may be fine mostly by themselves, not really hurting anybody necessarily. But at times, these abnormal cells can keep dividing and can form these little clusters that we call tumors. And that's where it's a growth we really don't want to see. And this could be dangerous because, again, the cells keep dividing unchecked. No one tells them to stop. They are incorrectly turned on in the first place. There usually is some kind of damage, some kind of mutation where they kind of get around the rules. They skirt around what's normal regulation of the cell cycle, and then keep going, and that would obviously be very, very bad. It can damage normal healthy tissue and can also spread around the body. That's what we call metastasis, and that's where you might hear this term primary cancer. Primary tumor can actually spread through the bloodstream to other areas of the body, and now you get this growth going other places as well, and not just confined to one area where the initial problem was. That would be even worse than regular primary cancer would be. All right, let's do some quick review questions and we're done. When does a cell need to copy its DNA? I'm not saying they copy one little recipe to make protein. When you need to copy the entire DNA molecule, that's when the cell is prepping to divide. So Remember, you need to copy your DNA first so that you have enough DNA to go around when the cell does eventually split into two, that everybody gets the correct amount of DNA. Why does DNA need to be copied before a cell divides? It's the exact same answer. It's just so that everybody gets enough. You can't have just whatever your starting amount of DNA was. I told you had 46 chromosomes. If you just split the cells right down the middle when you still had 46 and didn't make your copies, now at best you're getting 23 in each one. So you need to make enough DNA to go around. For number 3, place these words in size order smallest to biggest. Well, I know that genes are segments, little areas on chromosomes. And chromosomes are just those 46 smaller kind of organizational units of DNA. So chromosomes are just how DNA is sort of broken up. And the DNA is inside of a nucleus. So a nucleus is a big, that should say nucleus. Nucleus is a bigger structure. It holds the DNA. The DNA is divided up into chromosomes, and on each chromosome are typically a lot of different genes. How is the working of a cell cycle different in cancer cells compared to healthy cells? In cancer cells, it doesn't stop. Cell division is uncontrolled. It doesn't stop. It's turned on incorrectly. There's often mutations. With the normal cell cycle where you grow a little bit and then you make a copy and then you do your job again and then you divide and then you turn off and wait till later, that doesn't work. It's just divide, 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 divide. It doesn't stop. It doesn't stop. It doesn't stop. And that's really, really bad. That is it for today. Go fill in today's forum questions. Then you've got some worksheet papers and OneNote that I put up. I will put up another video on Thursday, and the work from both today and Thursday we do next Monday. That's the 11th at midnight. Now, the plan is still to have another extra help on Friday at noon, but as I mentioned in the email yesterday, I do expect to send an update to you at some point later this week with changes, so just keep an eye out for that. It sounds like we're going to have some kind of change to how we do extra help hours and stuff like that. Meantime, stay safe. I'll talk to you later in the week.